Today in this video we are going to talk about the various bacteria that secretes various exotoxins. So we'll talk about the various bacteria that secretes exotoxins. We'll also talk about what are those various toxins that are secreted by the various bacteria. We'll also talk about their mechanisms of the toxin action, how the toxin actually acts in our body, and we'll also talk about the manifestation of those toxins when they enter in our body. So generally classifying, here I have classified the bacteria with exotoxin in the 6 types. So those are those type of exotoxin which inhibits the protein synthesis, second which will increase the fluid secretion, and third which will inhibit the phagocytic activity, the fourth which will inhibit the release of neurotransmitter, another one those exotoxin which lies the cell membranes, and another are the super antigens that are causing the shock when they enter in our body. So first of all, let us talk about the exotoxins that inhibits the protein synthesis. So the exotoxin that inhibits the protein synthesis are secreted by the various bacteria. Various bacteria that secretes those type of exotoxin are Coronary Bacterium diphtheria, Pseudomonas aeruginosa, Shigella species, and enterohemorrhaginic E. coli. So first of all, let us talk about the Coronary Bacteria diphtheria. The Coronary Bacteria diphtheria secretes the diphtheria toxin, and I'm going to talk about Coronary Bacterium diphtheria and Pseudomonas aeruginosa together because their mechanism of action and manifestation little bit same. So Coronary Bacterium diphtheria and Pseudomonas aeruginosa. So Coronary Bacterium diphtheria secretes the diphtheria toxin, whereas the Pseudomonas aeruginosa secretes exotoxin A. And both of their mechanism of action are same. So they inactivate both the diphtheria toxin and the exotoxin A secreted by the Pseudomonas aeruginosa inactivate elongation factor 2. So these two exotoxins, diphtheria toxin and exotoxin A, inactivates the elongation factor 2, and those due to the inhibition of the inhibition or inactivation of the elongation factor 2, there is the inhibition of the protein synthesis. Now what is the manifestation in Coronobacteria diphtheria infection? So there will be the pharyngitis with pseudomembranous in the throat and the severe lymphadenopathy, and there is the bull neck appearances. Also there could be the myocarditis. So there will be the pseudomembrane formation in the Coronobacterium diphtheria infection, and there is the severe inflammation due to severe lymphadenopathy which causes the bull neck in the Coronobacterium diphtheria. Whereas in the Pseudomonas aeruginosa there could be the death of the cell host. Now let us talk about the Shigella species. The Shigella species secretes the Cigar Toxin. So by secreting the Cigar Toxin, it acts by inactivating the 60S ribosome by removing the adenine from the ribosomal RNA. Also the enterohemorrhaginic E. coli secretes the cigar-like toxin. That means the Shigella species secretes the cigar toxin, whereas the enterohemorrhagic E. coli secretes the cigar-like toxin. So both Shigella species exotoxin and the enterohemorrhaginic E. coli toxin acts by the same mechanism. They cigar toxin and cigar-like toxin acts by inactivating 60X ribosome by removing the adenine from the ribosomal RNA. So what are the various manifestations of infection by Shigella species? So the Shigella species secretes the cigar toxin, and those cigar toxin causes the GI mucosal damage and it causes the dysentery. The cigar-like toxin, the cigar toxin also enhances the cytokine release, causing the hemolytic uremic syndrome. And the cigar-like toxin enhances the release of the cytokine, which causes hemolytic uremic syndrome. So unlike Shigella, enterohemorrhaginic E. coli does not invade the host cells. So this is all about the various bacteria and their toxin which causes the inhibition of the protein synthesis. Now let us talk about the various bacteria with exotoxin, whose exotoxin increase the fuel secretion in our body. So those are enterotoxigenic E. coli, Bacillus anthracis, and Vibrio cholerae. So these three, the exotoxins secreted by these three bacteria will causes increased fuel secretion. Let us talk about their toxins, mechanism of action, and manifestation. So the enterotoxigenic E. coli will secrete the heat labile toxin. The heat labile toxin acts by overactivating adenylyl cyclase. So it overactivates the adenylyl cyclase, which increases the chloride secretion in the gut and the water efflux. So this works like this. The first, again I am going to tell you, the enterotoxigenic E. coli secretes two toxins. Those are two exotoxins. One is heat labile toxin, another is heat stable toxin. So both heat labile toxin and heat stable toxin secreted by the enterotoxigenic E. coli will increase the fuel secretion. First of all, let us talk about how heat labile toxin will increase the fuel secretion. The heat labile toxin will overactivate adenylyl cyclase and it will increase the chloride secretion in the gut and the water efflux. Whereas the heat stable toxin secreted by the enterotoxigenic E. coli will overactivate the guanylate cyclase, cyclic GMP, and it will decrease the reabsorption of the sodium chloride and the water in the gut. So the manifestation of the infection of the enterotoxigenic E. coli will be watery diarrhea. Now let us talk about the Bacillus anthracis. So the Bacillus anthracis will secrete the anthrax toxin. So the anthrax toxin will mimic the adenylate cyclase. That means it will increase the cyclic AMP, ultimately increase the fluid secretion. So we can see the it is likely responsible for the characteristic edematous border of the black hissure in the cutaneous anthrax, the Bacillus anthracis. So now let us talk about the Vibrio cholera. The Vibrio cholera will release the cholera toxin. The cholera toxin is the exotoxin which overactivate the adenylyl cyclase, cyclic AMP, by permanently activating stimulating G type of receptor. So it will increase the chloride secretion in the gut and water efflux. So it will ultimately causes the increment in the fluid secretion. So there will be the voluminous rice water diarrhea in the Vibrio cholerae. Now let us talk about the various bacteria which will inhibit the phagocytic activity. The bacteria which will inhibit the phagocytic activity are Bordetella pertussis. The Bordetella pertussis will release the pertussis toxin, which will inactivate the inhibitory G subunits of G inhibitory surface receptor, and their activation of the adenylyl cyclase would increase the cyclic AMP. So there could be the whooping cough. Whooping cough will be presented is the manifestation of the infection of the Bordetella pertussis. What happens is that the child cough on expiration and whoops on the inspiration. So this is the main feature of the whooping cough. Now let us talk about the toxin which inhibits the release of the neurotransmitter. Those are Clostridium tetani and Clostridium botulinum. The Clostridium tetani will release the tetanospasmin. So the tetanospasmin is the proteases that cleaves the soluble NSF attachment protein receptor. So it is the set of protein required for neurotransmitter release via the vesicular fusion. Also the same mechanism of action applies in the Clostridium botulinum. So Clostridium botulinum will release the botulinum toxin. So botulinum toxin and tetanospasmin both are proteases. So it will inhibit the release of the neurotransmitter via the vesicular fusion. So what will be the manifestation in the Clostridium tetani? So toxin prevents the release of inhibitory neurotransmitter from the Renshaw cell in the spinal cord, which will lead to the spastic paralysis and the trismus. So there is the spasm in the Clostridium tetani infection. Now let us discuss about the botulinum toxin manifestation. The Clostridium botulinum, that means botulinum toxin will, this toxin prevents the release of stimulatory signals at the neuromuscular junction, so there will be the flaccid paralysis in the case of infection with Clostridium botulinum. Now let us discuss about the various exotoxins which lies the cell membrane. First of all, let us talk about the Clostridium perfringens. The Clostridium perfringens will release the alpha toxin. So alpha toxin is the phospholipase that degrades the tissue and the cell membranes. So manifestation will be the degradation of the phospholipid, and there will be the myonecrosis in the Clostridium perfringens infection. There can also be the occurrence of the gas gangrene and the hemolysis. The Streptococcus pyogenes will release the streptolysin O. It is the protein that degrades the cell membrane. So Streptococcus pyogenes lyses the red blood cell and contributes to the beta hemolysis. Host antibody against the toxin ASO is used to diagnose the rheumatic fever. So we should not confuse this with immune complex of the procystococcal glomerular nephritis. Now let us talk about the super antigens causing the shock. Various super antigens causing the shocks are secreted by the Staphylococcus aureus and Streptococcus pyogenes. The Staphylococcus aureus secretes the toxic shock syndrome toxin. Staphylococcus aureus secretes the toxic shock syndrome toxin. So what happens is that the toxin secreted by the Staphylococcus aureus and the Streptococcus pyogenes mechanism of action are alike. So Staphylococcus aureus will secrete the toxic shock syndrome toxin, and the Streptococcus pyogene will release the erythrogenic exotoxin A. So they both cross-links the beta range region of the T cell receptor to MHC class 2 on the antigen presenting cell outside of the antigen binding site. So there will be the overwhelming release of the interleukin 1, interleukin 2, interferon gamma, tumor necrosis factor alpha, which will ultimately lead to the shock. So the manifestation of the Staphylococcus aureus infection or toxic shock syndrome toxin is toxic shock syndrome. So there will be, in the toxic shock syndrome, there is the fever, rash, shock, and other toxin cause scalded skin syndrome, that is exfoliative toxin, and the food poisoning. The Streptococcus pyogene toxin, erythrogenic exotoxin A, will cause toxic shock like syndrome. Also it causes fever, rash, shock, and scarlet fever. So these are the various bacteria with exotoxins. Again I am going to briefly summarize. So the various bacteria's toxin which inhibit the protein synthesis are Corynebacterium diphtheria, Pseudomonas aeruginosa, Shigella species, and enterohemerogenic E. coli. Those increase the fluid secretion are enterotoxigenic E. coli, Bacillus anthracis, Vibrio Cholera, and those which inhibit the phagocytic activity or phagocytic ability are Bordetella protosis. Those who inhibit the release of neurotransmitter are Clostridium tetani and Clostridium botulinum. Those who lyse cell membranes are Clostridium perfringens exotoxin and Streptococcus pyogenes. The super antigen causing shock are Staphylococcus aureus and Streptococcus pyogenes. So I hope you understood this topic with various bacteria with exotoxins. Thank you.